Okay, so we're going to continue on with what we've been talking about. On this is it's important that you pay attention, especially when we talk about the actual translation of the DNA genetic code from DNA to RNA, because and we're going to talk about it. RNA actually codes for actual amino acids, and you're going to have to use your slides. And I provided a tool for you to use to be able to translate RNA from DNA and to see what the actual amino acids are. So without further ado, we'll get started. All right, like I said, today we're going to talk about protein synthesis, because you're basically DNA is genetic code, but that it has to be it's broken down into like a protein sequence is what it actually codes for, and that is you. Protein sequence is your hair color, eye color, height, everything. Okay, genes in the DNA code, and just for just to make sure we're on the same page and you're aware of things. The DNA is what's in the chromosomes. So when you talk about chromosomes, that is the DNA. Okay, and so the DNA code for proteins that make your traits. Like I said, the protein sequence is why you look the way you are and everything you did and how tall you are and everything. Okay, and the DNA code can be likened to like a chef. They read a recipe to make a food dish. The cell reads the gene to make the protein, and the protein codes for whatever it is, eye color, height, all that stuff. These are examples of specific genes. The TAS2R38 gene, the TAS2R38 gene codes for you having a bitter taste receptor. And one of the interesting things I like to point out, and I probably will show you guys this at some point, because we actually, before the end of the semester, will perhaps have a legitimate reason. We'll have a legitimate reason to watch Jurassic Park, but actually talk about this in Jurassic Park. Uh, and a lot more breakthroughs have happened in genetic engineering. But what I like about a Jurassic Park is that the science is actually that they mention is actually can happen. And one of the things with the in Jurassic Park, they genetically engineered the animals to be lysine deficient. Lysine is one of the amino acids that animals that you need to be able to reproduce offspring. So without that, everybody would be ineffectively, all the animals would have been sterile. But then, things happen. Pretty much on the island, all the plants were full of lysine. So the animals started eating the plants, and then they were able to reproduce. So, but we'll hopefully we'll get to watch that. Okay, genetics of taste. You change the gene, change the protein, you change the particular trait. Okay, and when we get into genetic disorders and especially cancer, cancer is caused by genetic mutations, especially in the RNA sequence or DNA as well too. But and that can actually when the RNA actually codes for amino acids. So if one letter changes, sometimes that can change what the actual amino acid is, and so that could actually change the protein. And if you change the protein, you could actually change the trait. Like here, the trait for bitter taste. If you change a few of the letters out, as you see there, and we'll get to actually what that means. You can actually change the trait, and so you may not be able to taste. The person may not be able to taste bitter things like they should or whatever. How are proteins made? Proteins are made by putting amino acids together. Okay, made by putting strings of amino acids, and the place where it happens is the ribosome. Okay, if you and I guess I need to I'll show you next class, but I'll show you a picture of a cell. You can actually see the ribosomes. They're located on the endoplasmic what they call the rough endoplasmic reticulum, which is just outside the nucleus of the cell attached to it. And the ribosomes are these little, they look like little nodules, or they look like dots, but they're like little nodules on there. Okay, and that is the site where when the RNA comes back out of the nucleus after copying the DNA genetic code, then it goes and the amino acids are assembled on the ribosomes, and they're assembled in the letters are in groups of 3 called codons. Okay, so strings of amino acids become a peptide, and then the peptide chain, and then the peptide becomes a protein. Protein sequence is no joke. I mean, they have technology now to be able to break down the sequence faster, but you know, I knew a person that was his job, and it took him 2 years to break down the protein sequence from this plant. How are proteins made? Again, how does the cell know how to put the protein chain together? Obviously, it's from the DNA code. The DNA is like the blueprint, or like they were talking about with a chef, it's like the recipe. Okay, so one gene can be used to make one protein. So this could be the example of a gene, and by now, because we know, because we know how to do the base pairing, you guys should be able to tell me what the base pairing for this is. All right, DNA is too big and has thousands of genes. This is too important to leave in the nucleus. So that's why you have to, and we're getting into the two terms that you really need to be up on is transcription and translation. Okay, and if I transcribe something, like for to give you an example, back in like the 1500s, okay, they didn't have a whole lot of books, and one of the biggest deal is that there was a time before they had a printing press. So like the main book people read out of was the Bible. So what you had to do is, and that's what the priests and monks used to do who lived in a monastery, is you'd have a copy of a written copy of a Bible here, and you'd have a blank book of pages, and all it would do all day is copy from this book into this blank book, copy all the words. And that is what transcription is. So the RNA goes in, the DNA the double helix unravels, and then the single-stranded RNA, the RNA goes in there and makes a single-strand copy of the DNA. That's transcription. Now, we're going to get into both transcription and translation, but right now, if I transcribe something, it's just making a copy. So, and we don't need to belabor this, but you know, the bottom line you need to know is that RNA goes in and makes a single strand copy of DNA. And always remember, the reason it has to do that is because number one, DNA molecule is so big, okay, but number two, DNA never leaves the nucleus. Just like I was telling you about the archive section of the library. Okay, you can go in there and look at stuff and make copies of it, but you can't take it out. That's the way DNA is. Okay, so the DNA goes back out of the and I mean they have different terminologies for the same thing. The messenger RNA or mRNA is what goes to the ribosome, and the ribosome reads the mRNA 3 bases at a time, and that's what I'm saying. They call them triplets of codons. Okay, and then they take each codon and it corresponds with amino acids, and then they connect amino acids together. Okay, and here's a little animation. Okay, the first part was in the double helix, and now it went out to the ribosome. Okay, and I have this slideshow posted so you can go back and look at that. So we start with DNA. It's kind of the flow of information. We start with DNA. We go to RNA. Then at the ribosome, please remember that especially for quiz purposes, that the amino acid chain is assembled on the ribosome, and then after the chains of amino acids, which uh, get together or put together at the ribosome, they eventually become the full-blown proteins. Okay. All right, and this is just I've already talked about it, but we want to just hammer it home again. Transcription is rewriting or just making a copy. Translation is when you transfer that the code that was taken from DNA and is actually transferred into actual sequences of amino acids that lead to making protein. Okay, DNA versus RNA. Ad infinitum. You should know what the differences are now. Okay. Obviously, DNA has a deoxyribose. It's double-stranded. Has adenine, thymine, guanine, and cytosine. RNA has ribose as a sugar. It's single-stranded, and instead of having thymine, it has uracil. Okay, at this point, being quite honest, you guys should know that right now. Okay, when you look at a strand and you see uracil, you know it is RNA. Transcription is making an mRNA copy of DNA. The cell picks one gene to copy, and an RNA molecule is made from the unique DNA sequence. RNA then leaves the nucleus, like I was saying before, and transcription is makes the copy. You already know the pairing rules, so this should be easy. One of the things I want you to focus in on, though, is that when the DNA unravels, okay, helicase causes it to unravel, there's chemicals or nucleotides floating around, and that's how it covers down to make the single strand of RNA. Okay, so that's when so RNA in transcription just again is a copy of the genetic code, and since the double strands in DNA are complementary of each other, that's why you only need to copy one strand. And then again, translation is the changing of basically the DNA language over to the become chains of amino acids. Okay, so like I said, the ribosome is reads the mRNA code and assembles the amino acids into chains, and then those chains of amino acids become protein. But once again, the actual genes or the actual sequence that causes you to be who you are is that protein sequence. Okay. And this is we're just kind of reiterating it. Okay. The big picture: DNA holds our genetic information. Protein synthesis is a process, of course, of making proteins, and the proteins do the jobs in all our cells, giving us traits. And like I said, I wanted to mention again that what they're doing with the vaccine, several of the vaccines that they're giving us for COVID, is they're injecting that messenger RNA to tell the cells to build the code for the cells to build or to produce antibodies against the vaccine. So it's really genetic engineering. It's not the typical vaccine. Okay, protein synthesis reminders of what kind of you guys should at this point should know what transcription is. And then translation is just like if I'm translating from English to French, and if somebody says to me something in English, then I'll process it, and then I'll tell the person who speaks French what they said in French. So DNA is one language. It has the code. RNA copies it and then transfers it at the ribosome to become an amino acid sequence. Okay, and here's an example. We all should be able to, y'all should be able to do this by now. Okay, T, every place there's a T, you still put an A, but every place you would have put a T, you put a U for RNA. So TTG would be TTG would be AAC, GCT would be CGA. Okay, you guys, at this point, we should be able to make this happen. Okay, now this is the wheel and on your class assignment today. You're gonna have to be able to use this wheel to be able to tell what the different codons you're given translate into as far as amino acids, and it's easy, like GCA. Okay, GCA, you start in the center, so you go to G, then you work from the center out, so then you go to C, okay, and then you go to the outer wheel and you find A. So GCA would code for alanine. That's how you do it. And they'll tell you this: inner 30, first letter, middle ring, second letter. Yeah, just like that. Right. And so the amino acids in the very outer ring. Okay, so for example, AUG. Start with A. Okay. Start with the A. Okay. Then you go to the U, that's in the middle ring. And then you can find G. And that particular amino acid is methionine. And it's also, whenever you see the arrow, that means it's a start codon. And we'll talk about that. They have start and stop codons. Okay, when we see an arrow, that means it's a start codon. And if you see a circle, that means it's a stop codon. What will the rest of the amino acid sequence be? Well, let's see. Okay, so I mean, that's pretty much it. And UUA, let me show you what I mean by stop. So we're gonna start with, UAA. You're gonna start with U, then you go to A, then you go to A. So, and then when I told you when you see a circle, that's a stop codon. So, protein synthesis practice. That's what you're gonna do on your assignment. Okay, shortly. It's protein synthesis practice, and that's what I want you guys to work on. Now look, I can't emphasize enough. I'm giving you're gonna have enough time during the class period to work on this, and you should be able to finish it. It's just 10 kind of translations you have to do. Okay, there should be no problem with doing the assignment and hitting submit, and not having to be worried about turning it in 3 weeks from now. So that's what I'm looking for. Okay. And that's pretty much it. Okay. So I just wanted to make sure I go through the main things, key highlights, just to kind of review. The DNA is copied by RNA. RNA goes out of the nucleus to the ribosome, and that's where the amino acid sequence is assembled. And then chains of amino acids make up proteins. Okay. And the process of DNA being copied by RNA is called transcription. The process of the genetic code that RNA is copied being translated into or made into amino acid chains is called translation. All right, and thank you very much, and hope you have a great day.